In this video, we will understand some of the experiments that has been conducted so that it has been come to a conclusion that fatty acid synthesis are been uh, downregulated and fatty acid oxidation are been upregulated when there is a differentiation from the embryonic stem cells to the definitive endoderm occurs. So one of the experiment that they did is they analyzed the data that is publicly available of the human embryonic stem cells and this definitive endoderms, and they can find out that the process, and they check upon the gene ontologies of the process that is related to the fatty acid oxidation, and they have find out there is a there is a some fold enrichment of the process that are involved in the fatty acid oxidations compared to the case of this fatty acid synthesis in the case of or in this definitive endoderms cells. So from this they made a initial hypothesis that it's the fatty acid oxidation that has been upregulated, and the fatty acid oxidation has been upregulated in the case of this definitive endoderms compared to the case of these human embryonic stem cells. And from this they did some uh, specific analysis of the genes that are involved in fatty acid synthesis, and they can see that the fatty acid synthesis genes are in low amount in in the case of this HUE acid and H1 samples, and whereas these fatty acid oxidations are in high amount. Genes that are involved in the fatty acid oxidations are in high amount or are upregulated compared to the case of these embryonic stem cells. So this further supports their hypothesis that fatty acid synthesis are been downregulated in the case of this definitive endoderms when the cells are differentiated from embryonic stem cells to the definitive endoderms, and simultaneously these fatty acid oxidations are been upregulated when the cells differentiate from embryonic stem cells to the definitive endoderms. And they validate this their analysis using Western blot. In this Western blot, we can see that the genes that are involved in fatty acid synthesis are low in definitive endoderms compared to the embryonic stem cells, such as ACC1 2. So this substantiate their finding from the bioinformatics of the data that is publicly available, and they also check some of the they also check or analyze the bioinformatics of some of the enzymes that are bottleneck for this fatty acid oxidations, and they can find out that some of the enzymes that are bottleneck for fatty acid oxidation, such as CPT1B and CPT2 are been upregulated in the case of definitive endoderms compared to the embryonic stem cells. So this gives an indication that it's the fatty acid oxidations are been upregulated in the case of definitive endoderms compared to the embryonic stem cells, and they further validate their hypothesis using OCR of the samples in the presence of this ETO, and they can see that ETO is usually consumes these fatty acid oxidations so that the signals goes down whenever there is a high amount of ETO is there. So when in the case of this embryonic stem cells plus ETO case, they can see that there is a decrease in the fatty acid oxidations, and similarly when they check for this definitive endoderms plus ETO, they can see the similar decrease in the similar or higher decrease in the this fatty acid oxidations. So this and the other two controls are that these embryonic stem cells. So the embryonic stem cells in alone shows a higher signal, whereas embryonic stem cells plus ETO shows a lower signal. So this indicates that these experiments works according to the hypothesis that ETO actually consumes the fatty acid oxidations, and thereafter this OCR readings goes down whenever there is a ECS plus ETO and DE plus ETO are there. So this is just an experimental this is just a control experiment to show that ETO works, and their actual experiments data is shown on the left hand side, where they can see that when the OCR readings for the fatty acid oxidations in the definitive endoterms are high compared to the case of this ESC, where because the fatty acid oxidations that are high in this DE will be consumed by the ETO, and it gives a higher signal compared to the case of ESC, where compared to this DE the fatty acid oxidations are in low amount, therefore it gives a low signal because low amount of fatty acid are being consumed by this ETO, so it gives a lower OCR signals. So this validates their hypothesis that fatty acid oxidations are being upregulated in the case of this definitive endoterms, or during the differentiation, the reprogramming occurs in a way that fatty acid oxidations are being upregulated. And when it comes to the lipid synthesis, they check the lipid droplets through this Nile red dye, and they can see that in the definitive endoterms, the Nile red lipid droplets usually seems to be decreased compared to the case of these embryonic stem cells. And this is the statistical analysis of these images, fluorescence images. In this case, we can see that definitive endoderms has a lower fluorescence intensity about this Nile red compared to the case of these embryonic stem cells, and it is statistically significant. Data is also statistically significant. So this indicates that the lipogenesis process goes down, or fatty acid synthesis process goes down, and the fatty acid oxidation process goes up when the cells differentiate, embryonic stem cells differentiate towards these definitive endoderms. So this is some of the preliminary experiments that they did to support their hypothesis. And if you need to understand more about this detail, you can follow up for more videos on the similar topic, or you can also go to the reference that has been provided in the description. That's all for this video. Thank you for your time.